Every drug that is FDA approved today at one time was an experimental drug, obviously. And testis cancer is a unique exception. There are several other unique exceptions. When platinum came out, we did what's called a phase 2 study where, as Pat mentioned, 47 consecutive patients got a treatment where we expected to have a 5 to 10% cure rate, and instead it escalated eventually to an 80% cure rate. So we felt it would not be ethical, and the FDA agreed to ever do a phase 3 study where patients were randomized to chemotherapy with or without platinum. But that's not the reality of cancer medicine. Most of the time when we make an improvement, it's measured in months, or survival increments are measured in single digits rather than this walk on the moon type of thing that we get with platinum. So everyone that develops a new drug, David included, that has a new type of strategy, there's a rationale for doing it. And there are thousands of these rationales each year. There are a lot of brilliant people working in the field. And so you first test the drug in preclinical systems, and the preclinical systems give you an idea as to whether it's actually killing cancer cells. You can then test it in small animal models to see, get an idea what the toxicity is, and now you're ready to look at it in human trials. And if you see that a drug is having acceptable toxicity in humans and some degree of activity, you try to figure out what drug to be used in. So we don't do this so much with chemotherapy anymore. We do it with these molecular targeted agents. We find a mutation. The trouble with finding a mutation is that we have 2,000 genes in the human genome. There are 180 known mutations, but there are thousands of mutations that we find when we look for them, and most of them are passengers. So just finding a mutation and finding a drug that blocks a mutation doesn't mean it's going to be a Gleevec success story or a Herceptin type of success story. So it's used either alone or in combination with therapy. If it's safe and effective, unless it's like platinum and testis cancer, you're going to have to test it. And appropriately so, the FDA won't approve something based upon treating 20 patients and showing that certain tumors shrunk for a period of time. So we have to do the clinical trials, as Pat Loehrer mentioned. And the clinical trials is not no treatment versus treatment, it's standard treatment versus experimental treatment. The experimental treatment might be standard treatment plus the new drug, or the new drug instead of standard treatment. Now, you mentioned the financial constraints. We used to do studies in universities, and they would be run by university academic physicians, such as what you see on the panel. The studies now come from pharmaceutical companies. Let me rephrase that. The new drugs come from pharmaceutical companies. We're talking about $20 to $40 million for a proton center. That's cheap compared to developing a new drug. You're talking about hundreds of millions of dollars to develop a new drug. Many of them don't work. It is unbelievably expensive to do clinical trials. I think it's wrong how expensive it is. We were able to successfully treat testis cancer on a shoestring budget. Those days are long gone. So in order to do a clinical trial, it is very, very expensive. Industry runs these trials. They don't run the trial, but they develop the drugs that we use. Patients want something that's new and exciting. Doctors want some things that are new and exciting. That's the only way progress is made, is by looking at new therapy rather than the same old therapy. But it would be wrong to assume that every new drug that comes out is going to be a benefit. All drugs have side effects, but not all drugs have beneficial effects. So that's perhaps a shorter answer than you wanted with a long answer, but that's the way I would look at that.
Yeah, I was going to add, sometimes we find results that we just didn't expect. Again, when Larry and I were boys as interns and residents, if someone had a cardiac arrest, if they had an MI, myocardial infarction, and were hospitalized in the ICU, and you had an arrhythmia, if you had ventricular tachycardia, we would give them lidocaine. I mean, why wouldn't you? This is what you use to stop. We just did it. I mean, I have no idea who did this, but some guy came up with this trial called the CAST trial, the Cardiac Antiarrhythmic Suppression Trial. And they randomized patients who had MIs and had arrhythmias to receive lidocaine or not. It turned out that the people who got lidocaine had a worse mortality. They died more. So when we do these trials, sometimes we find out things that go against our our inclination, and that advances the field forward.